science professor, Mary Lauren Benton. I'm curious, how many of y'all want to go into medicine someday or healthcare in some form or fashion? I figured a lot of you be in this class. How many of y'all are interested in computers or the way computers are used in medicine? A couple of you. How many of y'all have noticed just how prevalent computers are becoming in medicine? If you've been to the doctor, you know, when you're a patient, when you're in the room with the doctor, you see, you know, they're looking at a computer, looking at your records. And that's just the most obvious example where we see computers in medicine. What's really cool is behind the scenes, we've got professors like the one we have here today who are using algorithms and computer science techniques and AI to better understand human health, to understand DNA and the way it works and how it impacts disease. So our guest today, Mary Lauren Benton, and her students, she's in something called biomedical informatics. And she's going to introduce her students. She's got a couple of grad students here with her, and she's got a presentation for you today.
So we're playing with the presentation. Maybe I can introduce one of my grad students, introduce themselves.
Hi everyone. I'm a fourth year PhD at the Department of Computer Science at Baylor University, and I'm Joseph. What I focus on for my dissertation is how to build gene models for predicting gene expression through DNA sequencing of people and understanding complex phenotypes and how to better kind of understand diseases that are leveraged or inherited through genetics.
Fourth year PhD student in the Department of Computer Science, and I'm Shri. And my work is to build like computer science deep learning models, which are graph neural networks, AI stuff, to make sure that we understand the chromatin interaction, like the 3D structure of the genome. So that's what
Let me ask y'all something while they're working on this. What are some, you talked about different diseases. What are some of the diseases that you all look at? I mean, you're not in the hospital, you're at the computer, but yet you're thinking about disease.
Right. So a lot of these diseases, it's a complex interplay between environment and genetics. And depending on what type of disease it is, it kind of varies, like proportions. Things like diabetes and then heart diseases and Alzheimer's and cancer are all things that are heavily influenced by genetics. So we study some of those, and in the past project, we also looked at alcoholism and also marijuana use and tobacco use in adolescents, and how that, whether there is a specific genetic element or component that kind of plays into that, and whether people are more susceptible to developing that substance addiction because of that.
So maybe someday people could find out, oh, I'm more at risk for addiction in some form or fashion, or I'm more susceptible.
Right. So like maybe you're more prone to having that, and maybe there's a way we can kind of have like an early intervention so that you don't fall into that, you know, and you don't have to like deal with that when it's already too late.
Now, when you both were in high school, did you know you wanted to go into computer science, and more specifically, did you know you might have an interest in applying to healthcare?
I wanted to be a doctor. I hated computers. I actually got a C or C minus in computer, I think. But after thinking about going to college, I was like, you know what? I want to do biology. I want to go to med school. And then I saw that how the amount of people that are all doing med school or thinking about doing med school in biology, I was like, I got to find something that's like a little bit niche, or maybe something that kind of puts me over other people. And I was like, bioinformatics or biomedical informatics sounds like something that's kind of cool. So that's how I got it.
Yeah. So for me, I did both the majors. I'm interested in like video games on one side and biology on the other. So yeah, I'm kind of in the best of both worlds right now, which is bioinformatics, which has computer science and biology.
So what's the coolest part of, I call it your job, it's not your job, but y'all are full-time in a lot of ways. What's the coolest part of what you get to do?
The coolest part is really thinking about like biology in like a non-traditional way, I would say. Like a lot of people think about biology, they're thinking about, I got to do all these experiments and all that stuff. But a lot of that we're doing is kind of finding patterns in the human biology, like genetics and physiology, like trying to understand things through more of a kind of a modeling approach, right? Would you say?
Those are deep things. Awesome. Well, it's nice to meet all of you. My name is Lauren Benton. I'm an assistant professor in the computer science department, but I'm really a biology enthusiast, a geneticist at heart. So when I was in high school, I was also really interested in medical schools. I knew I wanted to do something medicine related. But the longer I spent thinking about what it would take to get into med school and what it actually would like to be a doctor, the more I realized that the things that I were interested in were things that I couldn't do in the clinic. I wanted to not just treat patients, but to understand why they get sick and how to prevent that or diagnose that early, rather than just treating symptoms after they show. So I actually got into bioinformatics at Baylor completely by accident. I was late to sign up for the orientation session that I wanted, and I ended up in computer science. But somebody told me, hey, if you like biology, bioinformatics is just biology plus computer science. So if you also like puzzles, games, and logic, this is a really great way to marry those two. So I signed up for that major instead, thinking I could always change my mind if I didn't like it. But I ended up really loving it, and that was something that gave me the ability to think about questions in biology, what using computers to solve them. So I'm still involved in the study of human health, but I do this from a research perspective. After I completed my degree here in Waco at Baylor, I went to Vanderbilt in Nashville for my master's and PhD, kept in this biomedical informatics the entire time. So it's been something that I've really enjoyed. And then I came back to Baylor in 2020. So this picture up here at the top, I actually did my PhD defense in my slippers because it was in the middle of the COVID. But I just want to give you a little bit of an intro about what bioinformatics is, what I think it is, the science of analyzing complex biological data. So where I like to, when I say that we sit, is we are these unicorns that sit right in the middle of biology, computer science, and statistics. So we use data, methods, and techniques from all three areas in order to make sense of big questions. So we turn questions from classes like this one into problems that computers can. And the part of biology that's really interesting to me is genetics. This is a chart of the top 10 leading causes of death in the United States. Does anyone want to guess how many of the top 10 have a genetic component or some genetic risk factors? So your DNA sequence can increase your risk for these diseases. There's 10. How many do you think? Yeah. Okay. Very close. 9. The only one, unintentional. Everything else, there's some genetic risk factors or some component of your DNA that can make you more susceptible or can help to tailor treatments and other things. Understanding our genetics is really important to understanding disease. But there is a lot of information contained in the DNA sequence. So there have been a lot of projects recently, including in the United States, with all of this projects, but around the world, to collect as much DNA sequencing as we can from as many different people as we can in order to understand how that code relates to the traits that we observe. And the reason that we need computer science and algorithms to study genetics is because the human genome is really complicated. Anyone studied DNA sequence in their classes yet? Biology, perhaps? Yeah, excellent. So maybe you're familiar with the sequence. This is the set of characters that live inside every cell in your body. There are 3 billion characters. So that DNA sequence for a human is about 3 billion characters long. So this bookcase would reach floor to ceiling in this bathroom. That contains one copy of one human genome sequence. But every single one of you in this room has a copy of that sequence in all of your cells. I think it's pretty incredible. From a computer scientist perspective, that's also a lot of data. How many of you, this is the digital generation, right? How many of you think you are pretty good at typing? You can type pretty quickly on a computer. Handful? Do you want to guess how long it would take you just to copy down one human genome sequence if you're typing? Let's say you type 8 hours a day, 7 days a week, every day. This is your full-time job. How long do you think it would take? 2 days. do you think, Caleb? Depends on what's going to take more than 50 years to complete the job. So not quite a human lifetime, but we're getting up there, right? You start now, you'd retire, you'd finish and you'd retire. But your cells can do that process in a matter of hours. Crazy. But when we want to understand what kinds of patterns are inside of the sequence and how they relate to things like your risk for heart disease, cancer, or how we can tailor what drugs the doctor prescribes to best treat your specific version of a disease, we have to understand what patterns occur inside of the sequence. And we have to do that for millions of people around the world. This is not something that we can do by hand, even if we really, really want to. So that is really what got me interested in the combination of computer science and genetics. Not only do we have a really big DNA sequence, but if we were to take any pair of sequences in this room and compare any pair of people, we would find millions of small differences, probably somewhere between 4 and 7 million individual single character changes in that gene. But these small changes or genetic variants are actually really important for influencing traits. So those very small differences or changes in our sequence are things that can influence risk for disease or other traits like height, for example. So probably some combination of those small changes are why I am 5'4" and my cousin is 6'5". I will always be jealous. Right. So these small changes, we don't even need to understand just one sequence. We have to understand everyone's sequences and the things that make us similar and different. So as a computer scientist, I tend to think about the genome as a series of letters. I just want to give you a quick overview of what part of genetics my lab is really focused on. We've heard a little bit from my students. We know that part of that sequence codes for proteins. These are genes, right? So that DNA can be transcribed into RNA. Predicting that level is what Joseph's project is all about, predicting how the DNA can be transcribed in RNA. Then that's translated into proteins. I stick my arrow for DNA replication. That's our central dogma of biology. I'm really interested in how we can go from the same sequence with a very small set of genes, and we understand how those genes are turned on and off. So while a small portion of your human genome actually can generate those proteins, other parts of it are like switches that help to control what genes are turned on and when, maybe in response to different environments, maybe to tell a cell that it should perform a different function than a liver cell or a heart cell. Right? Maybe over time, like as you grow and develop, the genes that you need and the proteins change. So these gene regulatory elements can help to turn genes. And why do we care? Well, many of those small changes in the genome I was talking about that lead to different traits, most of those are not in genes. They're actually in these non-gene regions that are how the genome can regulate those genes. So they're actually not part of the genes. They're actually changing how the genes work. And so most of our changes that are related to traits or diseases, like I showed you on the previous slide, those are things that are related, that are in these non-coding portions of the genes. Those are traditionally very hard to study because there's so much more of it, and we're not entirely sure what all of it does. So one of the things that my lab is interested in is understanding what's happening in those non-coding genes. We know that some of these sequences, these short switches, they turn on genes. They activate or enhance genes. We call them enhancers. And this is an example of one for one of my favorite genes, the sonic hedgehog gene. This gene is involved in limb development and everything from the mouse all the way up to humans. And there are short DNA sequences that turn that gene on in the right time and make sure that your limbs develop appropriately. When there are mutations or changes in that enhancer sequence, they can cause disease. So even really tiny changes to the human sequence, just one character difference, can lead to what's called preaxial polydactyly. So you grow extra digits on your enhancer. So really tiny changes can have really big effects. So figuring out where these sequences are that can enhance genes is really important if we want to understand which mutations are going to have an effect on the person. So one of the things that my lab does we really need to use computers to understand where these elements are and how they work. So one of the things that we do is write computer programs and algorithms to take in a bunch of these DNA sequences from different people, and use these algorithms to figure out or predict where these enhancers actually are in the DNA sequence. And then when we know we have predictions about where these sequences are, we can also make predictions about which ones are likely to be if we make changes to. And so these are things that we're working on in the group. And things that are, I think, really important to train the next generation to do is to have these platforms from biology that understand what problems you want to solve. And then we'll figure out how to teach computers to help us solve them. Are there any questions? An AI model. I know you can have your teacher, like a chat GPT or something, maybe to make images or songs. Yeah. Excellent. So we can use that kind. Those are really built on machine learning models are essentially a bunch of statistics that help us to make predictions. We use them for very fun things like making images, generating text, creating music, but we can also use them to help us study biology. So we use machine learning and these AI algorithms to help us figure out which sequences are going to be enhancers and which sequences are. One of the things that we've built in the group is we can teach the computer, we give it lots of examples of what we think a real enhancer looks like. These are things that have been tested by biologists in the lab. And they said, yes, the sequence will activate the gene, will turn the gene on. And we give it lots of so these are things that you can test. They don't have any activity, they don't do anything to the gene. And so we take all of those sequences and we give them to these machine learning or AI models and say, here's what a real enhancer looks like, here's what a sequence. And here's what the fake enhancers look like. And here are their sequences. And we use that, we use those true and false examples to generate features or things that can help us distinguish between enhancers and non-enhancers. And then we train models, somewhat like the ones that you might be using, to make predictions about sequences. Are they an enhancer or are they not? And so that way, when we find new data that maybe we don't have the answer, nobody's taken it into the lab and actually tested it to see if it's working, we can put it into our machine learning classifier and make a prediction instead. And we found by doing that around 93 to 99% of the time, we can tell you if the enhancer sequence is true or false based on this model that we built. That's really cool because now we don't have to take every single sequence we want to test into the lab. If we want to make predictions, we can use our model instead to predict, is this an answer or is it not? And then if we wanted to use those predictions later, we don't have to go through the expense and the time of validating all of those enhancers. We can use these predictions to do other things like down the road. So one of the things that we're going to work on next is actually taking a lot of those predictions and then trying to understand what do changes in that sequence do to our prediction. So that's something that we're working on next by incorporating more and more complex models. You heard a little bit from the students, including even things that use some of the same structures as your challenge. I'm going to pause there. What I would like to do as an activity is show you how algorithms work. We're not going to get out our computers and actually program something, but I want to give you some intuition about what we need to do as bioinformaticians to translate a problem from biology into a problem that the computer can solve, a set of instructions for our computer. So what we're going to do is I'm going to have you all help me. We will become the programmers, and we will have Joseph go out and call for a little bit. He's going to be our computer. What we're going to do is I'm going to give you a couple options once he goes into the hall of things that he can draw from that way. You can pick what we want it to be, and then we're going to write the instructions for him to make that drawing. We're going to write the algorithm. and then we'll have them come back in and test our algorithm. This kind of process is exactly what we do when we write computer code. We have to decide, what do we want? What output do we want? What instructions are going to get us there? And then we test them.
Right? How much further right? So,
Got a few more.
okay. Draw a wavy line. Draw a semicircle in the bottom of the base. Okay. A wavy line from the bottom of the smallest circle on the right down to the bottom of the right horizontal line.
Oh, pretty good.
Okay. Draw a very small vertical line at top half of the bigger circle.
There's a lot.
Okay. Draw upside down curly bracket. Upside down curly bracket on top of the semicircle. You mean like this?
Yeah, but
Upside down, like.
Yeah.
Okay, hold on. So like. Here, you can right there. That's okay, I got it. Draw 6 dots in the shape of the rectangle in the small. Like that. All right, there's 6 dots.
Dimensions, so we should like do like around like I mean, I see the vision, but
There's nothing bobblehead. A really small balloon. Right? Very long arm. Clearly could play in NBA or something.
like it's like those books where it was who was blank, and on the front cover the head is always like
Yeah.
You know, it's interesting that this would be the time where if we were programming this, we would go back to debug. We would say, all right, we need to specify the size of the big circle and where it's located. And we could edit some things until we got our program giving us the output that we were doing. This is, again, kind of a silly example, but something that we do every day when we're trying to take our questions in biology and turn them into something we have to figure out how to take the complicated thing that we want, break it down into steps, and put those steps in the right order so that we can tell the computer exactly what we want and exactly how we want that output. Otherwise, our computer has no goals beyond executing our instructions exactly how they're written. And so we, as people in charge of our competition, that's our job to help translate our knowledge into something that the computer can do. And when we can do that, they're a really great partner for us to be able to solve big, complicated problems that we would not be able to do. But there's also jobs in the healthcare industry. So everything from designing drugs, like drug development, to generating AI systems for physicians, to understanding the basic biology, sort of what our lab does, and how genes are turned on and off. There are lots of potential jobs in industry and in academia. People are doing research, they're doing drug development, they're working in hospitals. Almost every portion of biology and healthcare system generates data. So when you have data, bioinformatics can help you to make sense of that. So you see versions of this job all over the healthcare and biology.
Dr. Benton, was it weird for you when you kind of started pivoting? You're like, oh, I always thought I wanted to, be a doctor, and then you started realizing maybe you didn't exactly. How did you process that? What was that like for you?
Yeah, so I think it was a little strange. Like I think Joseph said this, I didn't really like computers very much. I didn't know how to type. You had to keep one hand on the mouse at all times. Very slow. And so it was a lot of kind of changing what I thought computer science was. I thought computer science was spending long hours in front of the computer, writing code, probably in the dark, probably with a hood on, with a lot of Mountain Dew. Like I had a very specific image of what a computer science looked like. In practice, we're problem solvers. We take things and we try to think about them logically, and we use the computer as a tool to do that. So making that transition about what it means to do computer science and to be a computer scientist, I think kind of transitioning my mindset was really helpful for me. The computer science is the tool that I use to answer the questions that I work with here.
Does your research just stay at Baylor?
No, so we don't keep our research just at Baylor. The things that we do influences the way that we think about DNA sequences. So we publish our research in journals that are read around the world, and people use that information to help come up with their own ideas and experiments to test or hypotheses. So we help generate hypotheses. We also work with labs around the United States. So I think someone mentioned one of the other things we study in my group is alcohol use and alcohol use disorders. So we work with folks at Oregon Health Science University to study that, and they take those insights and apply them directly to the clinic. So the kinds of people that we're talking to when we give research presentations are people like doctors, pharmaceutical companies, and other researchers that then take that information and use it in their own work. So we work with people far outside of Baylor, and then we make sure that our work is disseminated far outside.
So is it fair to say someday there might be, you hope maybe someday there's new medicines or new treatments that your work could influence?
Absolutely. Hopefully there will be new treatments. One of the things that we start to see, especially for things like gene regulation, there have been advances in that area that are impacting real people right now. So there have been some new drugs developed for sickle cell anemia that use some of the techniques that I was talking about and use some of the knowledge generated by people studying these genes in the body. They're using that to treat people right now. So these gene therapies that are coming online are a direct result of the kind of work and wouldn't be possible without our bioinformatics and computers.
Is it expensive to go to graduate school?
Please. To live on while you study. So most of our graduate programs in the computer science department, our PhDs are fully funded. You actually get paid to do that research and to build that skill set for your future. That is something I didn't know when I applied to grad school, so I was very happy.
What could you do in high school that would get you ready if you wanted to do this career?
Basic knowledge of how to program, how to write an algorithm, I think can be a really useful skill set. But really just stay curious. The thing that I look for the most in graduate students and people that are successful in this area is because they care about asking why. They want to know why things happen. Keep that from people who are there to just get the grade and move on. If you really are interested in what you're doing and why, how things work, I think that goes a long way.